The early ancestors of all land plants were for a water-dwelling organisms. Simple land plants that show some of these adaptations are the liverworts. Terrestrial plants surrounded by air are in constant danger of drying. In order to avoid this, the upper surface of the liverwort thallid with an outer layer of cells which fit closely together and have thick walls. Aquatic plants are able to absorb water and minerals directly from their watery environment. Water and minerals enter the liverwort by means of rhizoids, which are thin walls extending into the soil from the plant's lower surface. The mosses are another group of simple land plants. They have adaptations which make them even better suited than the liverworts to the conditions of life on land. Like most land plants, the mosses have alternate generations, and like the liverworts, the dominant structure is the gametophyte generation. But the moss plant, instead of being a simple thallus, has structures which superficially resemble the roots, stems, and leaves of higher plants. Mosses do not, however, possess the specialized conducting tissues found in ferns and seed plants. The moss plant stands upright, and its photosynthetic tissue consists of small leaf-like structures. The blade is usually only one cell thick except in the middle region. The cell walls of the stalk are thickened so that the moss plant can stand upright, although it has no specialized supporting tissues. Leaf-like structures growing out of an upright stalk present a much larger area of photosynthetic tissue to sunlight than does a thallose liverwort occupying the same area. Moss plants, like liverworts, are characterized by largely undifferentiated tissue, but in some plants, a type of conducting tissue can be seen. A cross-section of a stalk of Nium shows several layers of thick-walled cells which support the stalk. The major portion of the stalk is made up of undifferentiated tissue that combines the separate functions of photosynthesis and storage. In the very center are thick-walled cells, some of which conduct water and others which support the stem. The stems of many mosses lack even this much specialization. Rhizoids occur at the base of the stalk and serve to anchor the plant and obtain water. Rhizoids are only simple filaments, and as water gatherers, much less efficient than true roots. Water, along with dissolved minerals from the soil, must pass through the plant by simple cell-to-cell conduction. This simple type of circulation is effective only for short distances. With only poorly developed conducting cells, the plant is limited in size. Mosses can easily reproduce by vegetative multiplication, and in fact, a few species seem to reproduce only in this way. Some mosses bear gemmi, which when separated from the parent plant, develop into new plants. Although dependent on occasional wet periods for the process of sexual reproduction, many mosses nevertheless occur in extremely dry environments, such as the surface of bare rock outcroppings. Here they are subjected to extremes of dryness and of heat and cold. Rock mosses commonly grow in compact tufts to conserve moisture. Their blades may roll tightly and clasp the stalk. When wetted, the plants quickly respond, opening up to present their full photosynthetic surface to the light. The remarkable resistance of rock mosses to environmental extremes may also be due to a difference in the nature of the protoplasmic contents of the cells. Sexual reproduction appeared early in the evolution of plants. The most primitive form of sexual reproduction involves the fusion of similar motile reproductive cells or gametes. In more advanced plants, one gamete retains its motile form while the other increases in size, loses its mobility, and stores food to be used by the zygote. The differentiation of gametes into two kinds, each with a special function, is the basis of sexual differentiation. In mosses, The sex organs which produce the gametes develop on the leafy stalk of the gametophyte plant. In some mosses, the male sex organs, or antheridia, and the female sex organs, or archegonia, are borne on the same stalk. In other plants, they may be borne on different branches of the same plant, and in many cases, the sex organs are borne on separate plants. In the mature male gametophyte plant, the antheridia are produced in clumps surrounded by a crown of blades or in buds along the stem. Filaments called paraphyses occur along with the antheridia and help to hold water and prevent the antheridia from drying out. Certain specialized cells inside the antheridia develop into small motile sperm. Swarms of flagellated sperm are released from the antheridia, but the sperm cannot swim to the egg unless water is present. Water falling on the plants may enable the sperm to swim to the archegonia born on nearby female plants. In some mosses, several archegonia, each containing one egg, are found on the apex of the leafy structure. In certain other mosses, the archegonia may develop on special short branches along the length of the stalk, which lies prostrate on the ground. Since they are more frequently submerged in water, the process of fertilization presents fewer problems than when the archegonia are borne aloft. Sperm must swim to the opening of the neck of the archegonium and then down the neck in order to fertilize the egg at its base. The sperm move directly towards the opening, almost certainly attracted by a chemical substance released from the archegonium. The sperm move along the chemical gradient to its source. The zygote results from the union of a male and female gamete. Each gamete possesses a single set of chromosomes and is therefore haploid. When sperm and egg fuse, the resulting zygote contains one set of chromosomes from each gamete and is thus diploid, or 2N. The fertilized egg is the beginning of the 2N, or sporophyte, generation of the moss plant. As the zygote develops into the sporophyte plant, the lower end or foot, buried in the tissues of the gametophyte, absorbs water and some food materials for the developing sporophyte. As the sporophyte plant grows, the archegonium that contains it expands. Finally, the sporophyte grows so rapidly that it bursts its chamber and grows upward out of the protective archegonium. It carries with it the top portion of the archegonium called the calyptra. The sporophyte grows a long stalk or seta, at the upper end of which a capsule or spore case is produced. In many mosses, the sporophyte plant contains chloroplasts and is thus able to manufacture a large portion of its own carbohydrate food. It is still dependent upon the gametophyte parent plant, however, for water and other substances which the gametophyte gets from the soil. The sporophyte plant is the 2N generation, and all of its cells contain 2N chromosomes, but the spores it produces are haploid and mark the beginning of a new gametophyte generation. Within the maturing capsule is a spore-bearing zone. All the cells in this zone are called the spore mother cells. Each spore mother cell undergoes two final divisions in the process called meiosis, which results in 4 haploid spores. When the spore case is nearly ripe, the calyptra is usually dislodged, and the actual lid or operculum of the ripe capsule is revealed. The moss sporophyte stands erect above the gametophyte, enabling it to have a wider spore distribution. The moss capsule possesses a unique mechanism to regulate the release of spores. Beneath the operculum, which is shed at maturity, circles of teeth called the peristome obstruct the mouth of the spored cavity. The teeth bend outward as the air becomes dry and inward into the capsule as the air becomes moist. This movement regulates the release of the spores, retaining them within the capsule during moist conditions which are unfavorable for spore dispersal. When the weather is dry, the teeth open, allowing the spores to drop to the ground or be carried away by the wind. The spores have a tough outer coating and are resistant to drying. They may be carried great distances by the wind. Those that fall in a suitably moist environment germinate and give rise to a new gametophyte generation. When the spore germinates, a branched filament of cells develops. This thread-like structure is called the protonema. The protonema is green, for its cells are filled with chloroplasts. Rhizoids, which lack chlorophyll, grow from the protonema and penetrate the surface on which it is growing. At this stage, the moss plant resembles some of the filamentous algae. The leafy moss plant begins on the protonema as a pear-shaped bud. The stalk eventually develops its own rhizoids and becomes independent of the protonema. To summarize the life cycle of mosses, the familiar leafy moss plant is the dominant gametophyte generation and is composed of haploid cells. The gametophyte forms an antheridia and archegonia, which produce the haploid sperm and egg cells. A sperm and egg unite to form a diploid zygote. The zygote develops into the mature sporophyte. The sporophyte is to some extent dependent upon the gametophyte. The mature sporophyte produces spore mother cells which undergo meiotic division, each giving rise to 4 haploid spores. The spores germinate to form protonema, which then grow into leafy moss plants, thus completing the life cycle. The moss plants are simple, but they have made adaptations which have enabled them to achieve a successful life upon the land.